So hi everyone, I'm Zev Ben Shachar, Director of Communications at CytoReason, and welcome to this joint webinar on the new collaboration between CytoReason and the Crohn's and Colitis Foundation. We have with us Professor Yehuda Chowers, CytoReason's Chief Medical Officer, and Dr. Alan Moss, the Foundation's Chief Scientific Officer. And moderating the discussion is Angela Dobes, Senior Vice President of IBD Plexus at the Foundation. Angela, over to you.
Thanks, Zev. This strategic collaboration represents an important milestone in the Foundation's commitment to play a larger role in advancing life-changing precision medicine strategies. Today, we're going to hear from two IBD leaders about the significance of this collaboration and its ability to ignite development of smarter therapies through use of IBD Plexus data to inform CytoReason's disease models. Once the presentations conclude, we then look forward to engaging the audience and invite you to participate in a question and answer session. And now I'm going to introduce our speakers. So first, Yehuda Chowers, Chief Medical Officer at CytoReason, a distinguished gastroenterologist and IBD thought leader. His research contributions to the IBD field earned him many accolades, such as an AGA Research Award and committee appointments, including ECHO Scientific Committee member. Yehuda also serves on editorial boards of several high-impact journals, including Gut. Yehuda's decades-long journey really serves as an advantage for this collaboration, underscoring the critical importance of understanding the history of IBD drug development to shape the future, navigate complexities more effectively, and chart a course towards better therapies. Our second speaker is the Crohn's and Colitis Foundation's Chief Scientific Officer, Dr. Alan Moss, an accomplished gastroenterologist and researcher who has been funded by organizations such as the NIDDK and Helmsley Charitable Trust. Alan brings extensive experience collaborating with industry, academia, and patients to push forward drug development activities and patient care. His deep expertise is really complemented by his passion for advancing patient engagement and research. Amplifying the patient voice is paramount to really unlocking the full potential that medical innovations can offer patients, and hence a unique vantage point the Foundation brings to this strategic collaboration. And so a big welcome again to our speakers, and Yehuda, take it away.
Thank you very much. Hi, everybody. So a great privilege to present in this webinar and to collaborate, especially with CCF as an organization. Great pleasure to collaborate with Alan personally. I'll start by presenting off CytoReason. Let me share my presentation, our overall approach and how we think about the problem of modeling complex diseases, and how this can contribute to relevant research problems. Okay. So CytoReason was founded in 2016. It's a company that is basically in the AI space. It has 130 employees. We collaborate with major pharma companies that you could see at the right-hand side. We are backed up by publications in high leading journals. Our basic technology is the disease model, and the disease model basically is a structure that combines big data, which is composed of molecular data, including single cell, bulk, genetics, and other data types, with clinical data, such as clinical features of the disease. In this case, we're talking IBD, and effect analysis of different treatments, etc., to allow for a comprehensive view of the disease space. We combine the data with knowledge, and the knowledge is composed of cell-specific language models, external database priors, data or insights from key opinion leaders, and all this is brought together by machine learning, with additional summary statistics from clinical trials, which is proprietary data, and molecular correlation structures and perturbation data that allow us to analyze these models, or the data within these models, in a comprehensive manner. CytoReason is involved in different stages of drug development, starting from target ranking at the target discovery phase, indication prioritization, um, finding subpopulations that will be liable to the treatment, generating classifiers and biomarkers for improved patient care and trial design. We have many models, as you can see on the left-hand side, 20 in the I&I space and 6 in the oncology space, and each model has unique constituents based on the data that is available in different comparisons. For instance, we compare disease versus control, and inflamed versus healthy, and inflamed versus not inflamed within the patients, etc. And in the case of ulcerative colitis, we have the number of treatments analysis available. Our data overall is composed of the public data, which I just shown you, and proprietary data. And the proprietary data is composed of data, partnering data, such as the ones that we have now, a major collaboration with CCF, customer data, and data generation to particular slots where the data that we already have doesn't give us an adequate answer. I will leave out the Plexus data because I know that Alan will talk about this, and I think he knows more about this than me. So what is the challenge? The overall challenge that we try to answer in the phase 2 trials is to increase patient recruitment and reduce screen failures. And we also try to support drug positioning for improved market access based on analysis of the data within the models. Basically, we try to bridge over 3 major data types. The first, shown in this triangle on the top, is the tissue data. The second is the blood data, which is the most available tissue for analysis. And we try to link these two to each other and to the third data type, which is the clinical data, from which we generate both from our present data and the public data sets and from real-world data such as the CCF data. So by bridging between these different data types, we're able to, or we're trying to provide the correlations between the tissue structure and molecular constituents with the blood molecular characteristics and with the clinical data available in the real world. How would we do that? Specifically regarding drugs, we could approach the issue of finding biomarkers for a particular drug in 2 ways. One way would be the drug-based positioning. Here we will leverage the data of clinical and molecular data from the tissue and the clinical and molecular data from the blood, and start by working on the target, characterizing its mechanism of action. This could be done with data that is provided by our collaborators or in a predictive manner on models that we generate for the drugs. After defining the putative molecular mechanism of action of the drug, we will positioning it on the disease network to see which part of the disease would this drug be addressing. And thereafter, this is projected into the population and stratified to see how many of which patient population is amenable to treatment by this particular drug. We will then generate tissue classifier for this particular drug and see how these drugs are reflected in the blood, for which we have pair clinical and molecular data in the blood of those very patients of which we use to characterize the tissue. The product in this case would be blood biomarkers on clinically annotated patients. The blood biomarkers would be for this particular drug. A second approach that appears slightly more complex would be what we term endotype-based positioning. What we will do here is use the same type of data, clinical and molecular data of tissue, with clinical and molecular data of the blood. Our approach here would be to divide or stratify the tissue into endotypes, say, for example, endotype 1, 2, 3, 4, correlate that with endotyping in the blood, then continue to generate a tissue classifier, generate tissue biomarkers for the endotypes, and then take the target, work up the target with its MOA, and position the target in the different endotypes. As you could see here, presented schematically, some of the types would be highly compatible with the tissue, with the drug mechanism of action. Other would be compatible, but to a lesser degree, and some would not be compatible with it at all. So what we would provide here are endotypes with varying degrees of compatibility with the mechanism of action of the drug. You will then translate the tissue stratification into blood stratification and provide blood biomarkers for target positioning and patient recruitment, but these will be biomarkers for the endotypes, not for the particular drug. So in general, just I would like to show shortly how we think about problems in a general manner. As you can imagine, there are multiple questions that need to be answered for different use cases, and we devise a systematic way to approach this. So for instance, we will have a use case here. Let's say, take for example, target prioritization. This use case will be addressed by answering a number of questions. So for example, a question could be, is my target associated with a natural disease course? These questions will be answered by criteria. These criteria, an example of a criteria could be an amplitude of change of this particular target along the disease trajectory. And the methodologies will then be, how do I measure these criteria? And an example of that will be an association with long-term tissue damage of the particular target. A common use case is the use case of evaluation of combinations. This is something that is now developing in multiple pharmas. And in this case, for instance, you will have different types of different drugs that should be positioned. And the question on the disease landscape, and the question that arises is, are these drugs additives, synergistic, or they could also be antagonizing? The way we would address this is to use or leverage the disease networks, assign and see whether there's an overlap or whether they're synergistic based on network tools, and then position the drugs into the effect of these drugs on the network as the effects of each drug separately, whether they're additive or potentially synergistic. One example could be in the space of IBD, is where we took will take now with CCF data, multiple data sets and integrate them together and stratify them. One of our approaches for stratification includes a cell-based stratification, followed by a stratification according to regulatory mechanisms. We will achieve this regulatory insight by adjusting for cell composition, leaving, so to speak, in the data, mainly the regulatory mechanisms that the data presents. We'll then combine these approaches, in this case, to create 4 endotypes, and then correlate these different strata with clinical features of the patients. These could be response or non-response, or response to surgery, benefit, long-term benefit from surgery, etc. Following this, when we already have these different strata that you see here on the left, we'll position either individual drugs or the drug combinations to see which endotypes would best fit the combinations of these drugs. I would like to end by showing how we actually interact with our customers. So we will take our disease models. We could actually have a project that is done by our scientists in collaboration with the scientist in our with a customer, and then which contains biologists, data scientists, and the customer leadership. The same data is also available via an API and via UI, where people who are not data scientists can actually approach and interrogate the data by themselves. Thank you very much. And I'll transfer this back to Angela. Thank you, Angela.
Great.
Comment?
Thanks, Yehuda. Alan, you're up next. Great.
Yeah, great. Thank you. Can you see the slides?
We can see it. You just need to put it in the presentation mode.
Yeah. Looking good?
Yes.
Awesome. So thanks, Yehuda. That was great overview of the CytoReason technology and models. But as everyone knows, great models need great data. And this is where the foundation comes in, and particularly IBD Plexus. The foundation sees IBD Plexus as a way to really leverage real-world data from clinical practice to accelerate discovery. Angela leads the Plexus program within the foundation, and on the team's behalf, I'm going to just share an overview of what the potential is with Plexus and how it will inform the CytoReason models. So, as a practicing gastroenterologist in IBD, many questions comes up in the clinic, and many questions for which there are no real good answers or partial answers to address them. And so that's really where experience from the clinic comes in terms of real world data. And I've just put here a couple of examples that come up probably in every IBD clinic every day around the world. Patients want to know, I've just been diagnosed with Crohn's or colitis. What's my course going to look like? Is it going to be a complicated course? Do I need to accelerate therapy quicker? Or am I going to have a benign course and maybe can have time to consider different options? If I do have a complicated course, what's the first treatment I should go on? Should it be a biologic first? Should it be an anti-TNF first, an anti-integrin, anti-IL-23? Or could I start with a small molecule or an oral agent first? And then probably most commonly, particularly in the larger centers, are I tried treatment X, I tried treatment Y, they failed. Why did they fail? And does that reason as to why that agent failed help you and me decide what agent I should pick next? Because thankfully, unlike 15, 20 years ago, we only had steroids or anti-TNFs. We now have several mechanisms of action available to patients and clinicians. But the challenge now is how to pick the right one for the right patient at the right time. So Plexus, as we alluded to, is really a platform developed by Angela's team at the foundation to really accelerate discoveries. You can see on this slide all of the data that's ingested into Plexus. It comes from both adult and pediatric cohorts. It shortly will be also adding in surgical cohorts. It comes from a variety of sites that are from diverse locations, geography, patient populations. And not only do we collect standardized questionnaires and PROs, we also ingest the electronic medical records of these patients. We have digitized images, for example, of histology slides. And there's almost 100 sites now participating in Plexus, informing or developing data that brought into the site or the platform. And it's a great resource for academics. There's over 150 papers have been published to date using the Plexus platform and the data. We have almost 30 partners in industry and biotech who use this to accelerate their discoveries. And we've invested a lot of time, effort, and money in generating transcriptomic and molecular data to collaborate and connect this to the patient's medical data and clinical outcomes. So that's just a flavor of what Plexus is and the potential for use with Plexus. And to dive a little bit deeper, particularly around IBD outcomes, you can see that Plexus allows us to integrate both clinical outcomes, like quality of life, for example, events in a patient's disease journey, with, for example, blood biomarkers, tissue biomarkers, and even stool samples and stool biomarkers. And it's unique, to my knowledge, in that respect, in terms of it can integrate all these variables, clinical outcomes, medical records, diagnostic codes, with what's happening biologically in the patient's blood, what's happening biologically in their tissue, and even what's happening in their stool samples. And so this is really an unprecedented resource and a source of data for informing models. Now, we realized, not so long ago, as the AI potential for accelerating discovery in IBD really started to pick up, that we needed to look out and find partners who had the technology, the right background, and the right ability to really make the most of the data in Plexus, and to bring some skills or technologies that we did not have internally to help accelerate this process. We've worked with many partners already on looking at, for example, digitizing images, but particularly for this partnership with CytoReason, we're very interested in looking at the patient disease journey and how we may use that to inform looking at the topics that Yehuda just discussed. Disease treatment failures, picking the right treatment for the right patient, disease enterotypes. All of those can potentially be really accelerated by combining the Plexus data with the CytoReason technology and models. This does not happen either without a lot of work within the foundation. A lot of the team are very heavily involved. We invest a lot of money in maintaining and supporting both the sites and the platform so that we can really provide the best resource available to our partners to accelerate all the discoveries that we want to accelerate in the IBD space. And some of the team members are here. This is what makes this work, and this is what makes this a successful platform for discoveries in IBD. And with that, I will just add, just to give you a flavor, we do so much work, and even as recently as at the recent DDW meeting in May in DC, you can see all great examples of where our partners have taken the data in Plexus and use it to publish academic and partner research, highlighting all the potential for this resource. So with that, I'm going to finish my presentation and happy to take questions from the group, and back to Angela.
Great, thanks, Alan. As Alan said, we're now opening the Q&A part of this webinar, so please feel free to type in your question in the Q&A, type in the chat, whatever is easiest. I do see we have got one question in the Q&A, and it's specifically around accessing Plexus. So IBD Plexus was really built for the IBD research community, and so there's many ways that you can access the IBD Plexus data set. I'll throw something in the chat. And then I think in particular, the one thing I want to highlight here is as we look to really give researchers access to this expedited data and biosamples, we've now also really begun to figure out strategically how can we partner with other amazing companies in the ecosystem like CytoReason to go further faster. So I'll put that in the chat, and you can through the link, you'll be able to understand who to reach out to if you're interested more in hearing about Plexus and of course our partnership and collaboration with CytoReason. Let me do that right now.
I just add, Angela, while you're doing that, at a high level, there's two ways to access. One is as an academic, you can request a proposal and submit it to the Plexus team and submit a hypothesis and a request to access that way. And then as a commercial partner, you can access to be a member or Plexus or have a relationship to access the data through those two. So there are two main channels for accessing the Plexus data. And now additionally, we're adding in these new partnerships like with CytoReason, where it's a combined model with the Plexus data and a models or algorithms provided by a new technology platform or a science platform to make the boast of actually combining the two.
Thank you. Okay, so I'll kick off, and I have a question for you, Yehuda. So one of the, as I think Alan mentioned this, you mentioned this a little bit too, one of the key challenges for leveraging AI in drug discovery is the availability of high quality and relevant data to really accurately understand the underlying disease mechanisms, right? Your presentation beautifully described the work that CytoReason is doing to leverage multimodal data, but what about the IBD Plexus data makes you kind of most excited about its ability to inform CytoReason's disease modeling work?
So many aspects. First of all, I think, that there are a lot of challenges, as you mentioned, with working with big data or multi-model data. And the time that it takes, the sheer time that these things take, is something that our technology solves and puts it together. Now, one of the problems that occurs when you integrate such vast data and you try to answer many questions is the size of the data or ability of sufficient data to do adequate validation. The foundation's data with its sheer numbers is something that allows you to actually address the problem of the validation in a very nice way, and a large independent cohort that allows you to perform validation. The second thing is that, as you know, or may know, is that the data that we obtain from the academic resources is data that is biased in a way, because most of these data are obtained from large academic centers. And those large academic centers and also clinical trials, which are performed in large academic centers, usually, they represent a relatively narrow fraction of the patient. So many patients are not represented. And for somebody who tries to develop drugs for the overall patient population, having data like data of the foundation data that is obtained from a variety of clinics, from a variety of physicians, allows you to actually be more precise in your ability to target larger and very relevant patient populations. Lastly, the foundation provides us with many types of data. So we have the molecular data, the RNA data, we have proteomic data, we have genetic data, and this will allow us to perform modeling that would be, let's say, more accurate, and will allow to address more diagnostic possibilities of the data. So all these things together really make the foundation's data pretty exciting for us.
And can I just maybe add context now, having looked at the data for some time with Angela's team, is it took almost 10 years with 100 sites to get to the level of where the data is in Plexus, right? If you were to start that now from scratch, it would be almost, I think, impossible to do it, firstly. And secondly, no one else has the network that the foundation has to actually go out and do this, because we're entirely dependent on this willingness of the sites to be involved, to collect the questionnaires, to collect the blood, to collect the tissue. And also, and I'm very conscious of this, we're also really grateful to the patients who take part. And the patients take part because they want to see, better ways to treat IBD. They want to see better diagnostics and treatments. And that really makes this so important, that the patients are invested in it, our sites are invested in it, and everyone is rowing in the right direction.
Thanks both. Alan, as you hit on kind of the why with the patients, and to really get to better therapies and treatments, we need to do this through clinical trials. It's no secret that there's barriers when it comes to clinical trials. And so as the foundation strives to lower some of these barriers, how can our collaboration with CytoReason help sponsors mitigate risk and facilitate faster recruitment into clinical trials, or any other areas that you think this partnership or collaboration could be fit for purpose?
That's for me, Angela?
Yes.
Yeah, okay. Yeah, so this is a big problem. I think both anyone in this space right now realizes that enrollment in clinical trials continues to decline, because the trials are all competing for the same cohort of people who are both meet the criteria for trials and are willing to be involved in a clinical trial. I think the number I saw recently, that the numbers are down to about 0.1 patient per month enrolling in some of these clinical trials, which is just, it's not practical or feasible to complete a trial in a timely manner and get these drugs to the regulators to decide on yes or no, we're going to approve this as a therapy. So that motivates certainly our patients. What we want to do with these kind of collaborations, like with CytoReason, is to say, okay, if you have a new therapy, you want to get into the IBD space, but you want to mitigate the risk for your individual product, this is a way to do it, because you can inform by patient stratification, by endotyping potentially. Here's the most likely group to benefit, and here's a group who's probably low likelihood to benefit, and that can help inform the trial rather than just taking let's just take everyone with Crohn's, try and enroll them in a trial like we've done for the last 15 years, that just not really, that's much more challenging going forward. The foundation wants to work with the sites to, provide the data to partners like CytoReason. We also want to work with our sites to get them trial ready, to get them shovel ready to be sites for clinical trials, and far more diverse sites than currently enrolling clinical trials. So they're the ways we're trying to make this more accessible for everybody.
If are you done, Alan? Yeah.
I'm done. I'm done, Yehuda. Sorry.
No, I didn't want to intrude. That's why I was asking. I was actually listening to you, being very satisfied because I concur with you completely with the points that you raised. I would just like to add another aspect to this. I think that the clinical trial field actually reflects real life. This difficulty to recruit enough patients to show statistically significant success rate that is demanded in clinical trials reflect the fact that in the clinical world, and I was a clinician actively practicing with patients, not in CytoReason, but in a public hospital for 35 years, it's very frustrating to see how we have this glass ceiling of response in the space of IBD. It's not an IBD specific problem, by the way, but this is our expertise. And when you see this relatively low rate of response, this really drives you to try and tailor the therapy to the patients right from the start. So if we would emerge out of these clinical trials, that A, would be easier for the pharma to recruit and reduce the failure rates because they're recruiting patients that have a higher chance to respond to what they're doing. And they come out with biomarkers that will allow the physicians to say, okay, you take this drug and you take that drug, you take it now or you take it later, etc. If we actually succeed in providing these type of tools, we've done a lot for the patients. And it's a joint effort of bodies like the foundation, which is a patient body, bodies like CytoReason, which is an analytic company, and the drug companies, which eventually produce the drugs. So I really think that this approach that has been called by many names, as personalized medicine and precision medicine, is something that is an unmet need in this area.
Thank you. Speaking of the glass ceiling, we got a question in the Q&A, really talking about stratifying patients, whether that be biomarkers, etc. How flexible is CytoReason's platform when stratifying patients into different groups? For example, within a flare or medication failure success, presence of fibrosis, perianal disease. Yehuda, can you talk a little bit about that?
With pleasure. Again, I think that the foundation's data will support us in this effort because eventually when you stratify your patients to molecular data to different clinical characteristics, be it a disease phenotype or a response, non-response to a given drug, or a stage of the disease, which is essentially what you were asking, there's a limitation eventually in the sample size of the patients that you're sampling in order to generate biomarkers or patient subpopulations according to these different subtypes. And indeed, I think that this disease heterogeneity is the cause of this glass ceiling. So from the computational point of view, we're extremely flexible. We have novel algorithms that we've developed. Some of them are published to define disease natural course and to associate disease characteristics with specific molecular features. The limitations will come mainly from the sample sizes, mainly from the accuracy of the annotations of the cohorts. So getting data like the foundation has, which is very accurately annotated from the clinical point of view with ample molecular and other types of data, as Alan alluded to during his presentation, will really progress the field.
Thank you. We're still on the roll with clinical trials. Alan, this is a question for you. Can you generally summarize some of the key barriers to greater clinical trials engagement?
Yeah, so before I was in this role, I was a site PI for 15 years. And so I can, from that perspective, tell you, the barriers that I saw and what the patients referred back to me. And this is, I think, not unique to my experience, is that firstly, the trial design is often overly restrictive in terms of who's eligible, who's not eligible, and overly cumbersome in terms of how many times the patient has to do a colonoscopy, give a stool sample, do a blood sample, and so forth. So I think trial design is one major barrier. And that feeds into things for patients like, do I have to come back to the hospital 6 times in 6 months for all these procedures? Who's going to look after the kids? Who's going to look after my parents? I got to take time off work. So the field as a whole is trying to move towards more remote clinical trial models. But in the IBD space, we're still heavily dependent on trials that are all about having a colonoscopy, doing a blood sample, doing a stool sample. So I would love to see sponsors and regulators allow more flexibility on these kind of remote measurements and not just having at the site. And I think there's also with patients some concerns, distrust about, well, am I in a experimental group? Like, is this trial, is this treatment tested? How is it tested? Am I a guinea pig is often a question that comes up. And we got to do a better job explaining to patients that these agents have been through multiple realms of toxicology, safety study, and volunteers before they ever get to the clinic, where we're saying maybe this might be a good option for your IBD. Would you be willing to try it out? So that's a short way of thinking about many complex areas that, in my view, are the barriers right now to enrolling, certainly in the US.
Thank you. A question for both of you. We have an audience member who would love to hear what are the 2 to 3 biggest problems you hope Plexus and/or this collaboration could solve for the IBD community.
You want to start, Alan? Do you want me to start?
You go ahead, Yehuda, and then I will finish.
Okay. So I think that we will actually, I think, through this collaboration, create a platform. It's not a singular solution, but create a platform that will allow to position patients in bins of response to the different drugs and test, often in a comparative manner, or rather in a complementary manner, what would be the right sequence or drug sequencing to treat them based on their biological characteristics. So I think that the ability to do this in a systematic manner on an equal background is something that is really missing today, because Alan has already alluded to this issue. Which drug should I start with? It's always this balance between the price, the therapeutic price, I mean, not the money, the therapeutic price that you're going to pay with a aggressive type of therapy that has a robust mechanism of action on your immune system versus the safety of the drug. What kind of physiologic advantage are you going to gain by doing that? If you have an overview of the disease landscape, it's easier to take these very basic therapeutic decisions. This reflects on drug development immediately. When you're developing your drug, if you're able to characterize your drug in the landscape, in the disease landscape, then it's easier for you to extrapolate and answer practical questions like patient recruitment and screen failures. But similarly, what is your added value in this landscape of developing drugs? There are today many drugs. I was in the field even before we had anti-TNF, Alan. So, so it was even narrower at the time. So I think that this ability to create a landscape, the disease landscape, with all its different angles, the disease phenotype, the disease natural course, the response to the various drugs, the response to surgery, all in one platform that allows you sort of a universal overview, to me, allows to answer many questions. And this can only be done with a combination of good technology and good data. And I think that each one of the partners to this collaboration gives a very complementary aspect to this investigation.
Yeah, and I will add to Yehuda's point that that's exactly how the conception for Plexus came from an adult cohort called Spark, which was really about precision medicine and picking the right drug for the right patients. As it's evolved over time, the potential is now even greater. I think we're now be able to look at things like drug safety, right? Are there adverse events in cohorts that are unexpected or occurring at greater events that we would previously have thought to occur? The second is biology. We're now seeing as the incidence of IBD increases in newly industrialized countries, we're seeing much more different flavors of IBD develop, and understanding the biology of why that is and what are the triggers in India or Brazil versus the triggers in Israel or in the UK is also really helpful to understand. And hopefully some of that information about biology will then lead us to thinking about how could we prevent IBD in at-risk populations. Now, they're big goals, but I think the potential is there, and this one focus on treatment decision-making and precision is really where the CytoReason team can help us with initially.
Thank you both. I'm going to shift gears a little bit, talk a little bit, ask a question that's more maybe operationally focused for all those data scientists on the call and those who have hands-on experience with data. What I have heard is data science teams often dedicate significant time and resources to organizing and preparing data to create data sets suitable for AI learning, right? This process can be time-consuming, challenging, hindering the ability to really generate valuable insights effectively. So Yehuda, how can CytoReason's offerings help organizations reduce the time and effort required to generate these insights and make these large multimodal data sets manageable?
So that's a definition of a model. The model is pre-computation and pre-organization of many data sets and many data types in a validated QC, Q8 manner. It's an industry. It's not an academic institution. And we undergo continuous scrutiny by our collaborators, which are very picky, and there's a lot at stake. So what we produce has to be at high quality. So all this part of organizing the data, and I completely agree, it's a very time-consuming, labor-intensive process. All this is done in the structure of a model. This is exactly. So if you, sorry for advertising here, if you buy our models, you get this pre-processed, and you also get all the reports of all the quality tests that have been done on this model.
Thank you. Alan, the foundation recently published our Challenges in IBD 2024 document. And as I read that and I reflect on this sequence of the IBD journey, including the preclinical stage, cycles of disease activity, remission, and restoration of normal function. Like, what are you most excited about for the future of IBD and opportunities to collaborate with companies like CytoReason?
Yeah, thanks, Angela. And so for those interested, maybe we can put it in the chat after that document. There is one review there just on precision medicine, and also one on future technologies in IBD. And so this collaboration would really appear in both as an example of where we're going. So you mentioned two topics, and I think what excites me most right now is, and I hope would excite you to also, to think about how could we prevent IBD? If we do know, for example, in Crohn's disease, there are at-risk groups who are much higher risk of developing Crohn's disease, and within families, there are often many siblings or family members involved. With those, is there a test we could do to identify who's at risk, and then think about an intervention to prevent the onset of Crohn's disease, like they've done with type 1 diabetes in children? On the far end of the spectrum, we have focused a lot over the years on getting people into remission, but we know from patients that remission means different things to different people. It may just mean I'm having less symptoms than before versus I feel back to normal. And so we kind of pioneered this concept of restoration, which is really pushing patients beyond remission into restoration of normality, getting functioning back towards normal, physiology back towards normal. And we're hoping with some of these collaborations, like, for example, the enterotyping work that Yehuda referred to in his presentation, would sort of give us insights into what that would look like and what is the biology to get us there from just clinical remission to physiological restoration. So I think both of those ends of the spectrum really excite me right now.
I would like to add that I fully agree with Alan that with the deeper understanding of factors that are associated with disease globally, we may be able to get closer to preventing the disease. And I also agree with the fact that understanding trajectories is something that allows you to refine factors. And the key to understanding trajectories is having patients at different points of the trajectory. And because we can't afford to wait for the 15, 20 years that it takes to understand the patient journey of, patient just started disease and underwent like 10 types of different treatments and surgery. You would like to develop a technology that allows you to take transactional data and turn it into a trajectory. And all these things, which is another thing that we do. But I agree completely that the future of IBD is in trying to identify the disease early, either preventing it or halting its progress during the years where tissue damage accumulates and complications occur.
Amazing. For those that are, on the call that are more familiar with IBD Plexus data, maybe they're users of the data. Yehuda, I had a question for you to just share a little bit more with the audience around kind of, again, how you guys can help maximize this value of IBD Plexus data. So I know one computational approach developed by CytoReason that I believe will help maximize the value of IBD Plexus data is its ability to take bulk or average gene expression data and estimate the cellular composition of the tissue. Can you share a little bit more about the benefits of that, knowing that we have a lot of bulk RNA sequencing data available on tissue samples through Plexus?
Sure, with pleasure. So the technology you're referring to is called deconvolution, which is basically a statistical method to take advantage of the bulk RNA and by computationally estimate what is the cell composition of this in this bulk RNA. Today, with the advent of single cell data, the ability to generate signatures of particular cells, etc., is something that is getting better and better. And Professor Shai Shenor, who is one of the founders of CytoReason, was actually one of the early developers of statistical deconvolution for determining cell composition. And the advantage of bulk, for instance, over single cell is that single cell indeed provides you a deep understanding of the biology of the cells that you can actually sequence, but you're sequencing much less cells than what you have in the bulk, which contains all the cells that are in the tissue. So these methodologies are actually complementary to each other. And indeed, we use both types of cell sequencing in our analysis, both the single cell and the bulk with the deconvolution.
Thank you. So I don't see any more questions in the Q&A or chat. Maybe one just came up. No, I don't. Correct me if I'm wrong. Anyone else sees any? If not, I think if no more questions, I think we can close out the webinar. Before I do, I just want to reiterate one comment I made earlier. We built Plexus for the IBD research community. We believe that this strategic collaboration with CytoReason will allow the entire community to go further, faster, to do better for patients. And so appreciate the time you took today to learn a little bit more about this collaboration. And of course, if you have any questions after the webinar, reach out. So with that said, thank you again and enjoy your day. Bye.
Thank you
Thank
very
you,
much.
Yehuda. Thank you, Angela.
Thank you, Alan. Thank you,
Thanks,
Angela.
everyone. Bye.
Bye.